So when last I left off, we were talking about the patterns of natural selection, and there are three basic kind of patterns that you're going to see in a population when natural selection is working on them. There's directional selection, and directional selection you can see actually in this middle two sets of graphs here. Directional selection is when the average in the population, and this is the original population right here, this little bell-shaped curve, when the average shifts one direction or the other. And in this case, it's shifting to the right. And that's because what these little arrows are showing is that individuals on this side of the graph are not surviving to reproduce, and the ones on this side of the graph are surviving to reproduce more. And what happens with that is that you get a shift to, in this case, the right side of the graph. And so that's evolution. The range of phenotypes has shifted over to the right. Okay, so imagine that this is a gene for height, and the short individuals are not reproducing as much as the tall individuals. That's kind of what it would be like. The second is stabilizing selection, and that's this first column over here. And with stabilizing selection, it's the ones in the middle that are doing the best. The ones on either extreme in the population, either the very, if we were talking about height again, the very short and the very tall, they're not doing very well. The middle height, that would be the one that would be doing really well. That's stabilizing selection. If you notice, the average stays the same, but the amount of variation in the population has decreased a lot. The last one is called disruptive selection, and with disruptive selection, you have the two extremes, the ones on the edges doing really well, and the one in the middle not doing well at all. So you end up getting this bimodal distribution, where, again, if you think about height, the very short ones and the very tall ones are doing better, and the ones that are average are not doing well at all. So those are the three general patterns. Let's get some actual real-world examples for each of these, starting with directional selection. And we can see directional selection on turkeys. Now, around Thanksgiving, everybody wants a nice big turkey with a lot of white meat there in the turkey breast. And so farmers have responded to this by only allowing the turkeys with the largest chest muscles to be the ones to reproduce. And so in the wild, the population might be like this in the gray, but in the farms, they're selecting against the ones with the smaller chest muscles, only allowing the ones with the larger breast sizes to reproduce. And because of that, we get this shift over to the right-hand side. That's directional selection. Very interestingly, because these breast muscles are so large, turkeys cannot mate with each other anymore. They require a human to transfer sperm into and over to the female so that the eggs can be fertilized. So kind of cray-cray. The next one is stabilizing selection. And we see stabilizing selection in humans. Human birth weight is a very interesting thing because the vast majority of babies that are born in a narrow range of birth weights. We see, like 90% of babies being born between 6 pounds and, like 9.5 pounds. The vast majority of babies are born in that. That's strange. It's a very much narrowing of what we would expect to see if there weren't this kind of pressure, where large babies are not born very often because anybody who in the history had a large baby, the baby probably died or the mother probably died. And anybody who had a baby back in history that was of small or low birth weight, the babies tended to pass away or die because of complications associated with that. So human babies with low birth weight or high birth weight are vulnerable. And so because that trait has been passed down, the ones of middle weight being the most successful, humans tend to be born in the middle weights and higher than expected levels. The next one is disruptive selection. And disruptive selection is, again, the the selecting against the average, and we can see this really nice example of this in some fish. Very specifically, we're talking about a species of salmon here. And so you might expect a normal population to have the gray-shaped curve here, but in fish, what we have is, especially this species of fish, the salmon, we have a bunch of fish that are small and a bunch of fish that are large, and the ones in the middle, there's hardly any of them. So there's a good reason for this, and that's because coho salmon are territorial. It sounds a little strange, but once those fish swim up that stream, the males fight for territories. And the larger the fish is, the more likely it is to get the territory. And the females are attracted to those large fish that get the territories. But the small fish, the small coho salmon, those are the ones that are called sneaker fish because they actually sneak up and fertilize eggs before they're detected. And so the males are busy defending against these medium-sized fish or some other bigger fish for the prime territory. And these little sneaker males come in and they fertilize the eggs, unbeknownst to anybody else, really. So that's disruptive selection. We also know that selection can act on very complex traits. Complex traits like behaviors can be affected by natural selection. And there was a really nice experiment done, very, very elegant experiment done on rats and rat intelligence. And so, you know, the classic where you put a rat in a maze and it tries to find its way through, making the fewest mistakes. Oop, there's a mistake. So it goes back and it tries, oh, it's a mistake there. Tries to find its way through the maze, right? Oh, getting stuck. So what they did was they counted how many mistakes that these rats made going through the maze. And turns out that the lab rats were all at the very beginning, were all very, very close to making about the same number of mistakes. However, there were a few of them that were a little bit slower at getting through that maze, and there were some of them that were a little bit quicker. And so what they did was they mated the ones who were quicker together, and they mated the ones that were slower. They mated those together with each other. And in the next generation, in generation 2, it turns out that the ones who had quicker parents were a little bit faster at learning that maze than the parents were. And the ones that had the slower parents, they themselves were even a little bit slower. This got pretty dramatic after generation 3, and after 6 generations, we had some slow learners that were really, really slow, and the quick learners that were really, really quick. And so what we see here is that that we've got changes in allele frequencies, because let's face it, intelligence is in part due to genetics, and then you can see how easy it is to select for those traits. In this very, very complicated trait of how many decisions and how fast you are getting through a maze. There's other complicated traits that natural selection can also work on. So the really interesting example here is wings. And what we know about wings is that if you have a tiny little wing, like a little nub, tiny little nub of a wing like you see right here, that's not going to help you fly. And so how can you get from a tiny little nub of a wing to having these really quite beautiful and amazing wings that can help you fly in this just beautiful way? And the way that this happens is something called a functional shift. I forgot to bold this for you, so there's your term, functional shift. And a functional shift is where a structure starts out having one function and evolves into having another function after it gets to be a certain way, in this case, a certain size. So these tiny little nubs, they don't really help with flight, but they do have a function. And that function, it says right here, is thermoregulatory, which simply is a fancy word for saying that it helps regulate their body temperature. Those little nubs, when it's really hot out, it helps get rid of excess body heat. And when it's kind of cold out, if they go sit in the sun, it helps them actually absorb more heat from the sun. So thermoregulatory. So the bigger these little nubs became, the better the thermoregulatory process was, the benefit was, up until a certain point. But of course, once you get to be a certain size little wing nub, a little medium-sized wing nub here, you actually start to get a little bit of lift when you're jumping. And so it's like a sort of gliding thing going on. And so once that gliding thing comes into place, now evolution is going to favor having true flight. And this takes a while to happen. It's not like it happens overnight. It's going to be many, 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 many generations for this to happen. It's whatever slight little improvement, however small, is going to be selected for in the case of flight, because that's going to get you away from your enemies and so forth. So natural selection really can work on complex traits. One of the most complicated traits of all is the human eye. And we've already talked a little bit about eyes. In humans, how we've got this blind spot and everything. There's other problems like, that retina can actually become detached and so forth. So it's, eyes are not perfect. But how can you get this very, very complicated thing that is an eye, human eye or reptilian eye or any kind of eye really, from something that's really, really not very complicated? How do you do that kind of thing that we're talking about to get to something that's really complicated? And it's definitely a stepwise process. You're not immediately going to just evolve an eye. So the first stage here is to have some cells that are sensitive to light. We call those pigment cells. And if you think about it, there are lots of cells that are sensitive to light. Your skin cells are sensitive to light. They change color. They make more melanin when they're exposed to light. So we have those. Even bacteria have in them chemicals that are sensitive to light. And so it's not a very big jump to think about having these pigment cells located kind of on the face. Now, by having a little indent here, this little cup here, what happens is you start to get a little bit of information about shadow. I'm going to show you this video. Hopefully it'll work. I don't usually show you videos in my lecture, but I'm going to do it this time because this has such a good explanation. So let's go ahead and take a look.
Valerie Young's retinal tear is just one example of imperfections in the design of human eyes.
Liam Neeson.
Another occurs because nerve cells and blood vessels evolve to lie in front of the retina, where they interfere with its ability to form sharp images. It's like trying to take a picture through a foggy piece of glass. And the optic nerve itself evolved to connect to the brain through a hole in the retina. So the eyes of all vertebrates have a small blind spot, right near the middle of the visual field.
Evolution starts with what's already there, tinkers with it and modifies it, but can never do a grand redesign. So even the eye, with all of its optical perfection, has clues to the fact that its origin is of the blind process of natural selection.
Darwin believed that what he called an organ of extreme complexity, like the eye, could evolve by small steps, given enough time. Any trait that improved vision would aid in the search for food, or a mate, or in the avoidance of predators. So natural selection would most certainly favor those traits.
And what Darwin was able to do was to point out that you might think in logic that it's difficult to imagine a set of intermediary stages between the simplest little spot of nerve cells that can perceive light to a lens-forming eye that makes complex images. But in fact, these intermediary forms do exist in nature.
At the University of Lund in Sweden, zoologist Dan Erik Nilsson has developed models to show how a primitive eye spot could evolve through intermediate stages to become a complex human-like eye in less than 500,000 years.
Yeah, I've been interested in eye evolution for a long time. And in particular, I've been interested in the question of how long time it would take for an eye to evolve.
Nielsen envisioned a sequence of stages by which a flat patch of light-sensitive cells on an animal's skin could evolve into a camera-type eye. As a first step, nature would favor any changes that made the flat patch more cup-like.
As soon as you created even the slightest depression in the center, it means that the edges of the cup will actually shade light from parts of the environment. And of course, all the light-sensitive cells in this little cup, they won't measure light in exactly the same direction. So already this cup has some pictorial information.
Another model demonstrates what a primitive cup eye can do. The brightly lighted skulls cast an image onto a translucent screen Nielsen installs at the back of the cup to act like a retina. But the image is not at all well defined. The cup eye can do little more than detect movement. This kind of eye can be found in nature today, in flatworms. Their eyes evolved no further. In their environment, that's all they needed.
But if the animals need to move faster, or evolve to become fast predators, or to see other fast predators, then the construction needs to be improved. And one way of doing that is to constrict the opening, to make it smaller.
That's just what happened to creatures like the chambered nautilus. Over thousands of generations, natural selection favored those with slightly more constricted eye openings, which focused light more sharply. This worked well, up to a point.
Since this strategy of making a sharp image also has the drawback of creating a very dim image, it's not very popular in the animal kingdom. And there is an alternative solution, which has become very popular in the animal kingdom, the solution that we use in our own eyes, and that is to put in a lens.
Nielsen's model lens uses two thin layers of clear plastic. He can inject water in between them to make the plastic windows bulge out like a convex lens. This mimics what natural selection might have done over a few 100,000 generations, favoring animals with a rounded, transparent layer in their eyes that caused light to be focused more sharply on the retina.
So we can make it gradually from no lens at all and just continue to inject more water, making the lenses bulge more and more, and the image becomes gradually sharper and sharper. So we can go all the way gradually in very small steps from a simple pigment cup eye, which has barely got the ability to determine the direction of a light source, all the way to a complete camera type eye of the same type as we have ourselves. And that is really exactly the way eye evolution must proceed.
And I'm just going to go ahead and stop it there. So I really like that video, and I think that it's posted on Blackboard, but I'm not 100% sure. I'll look into that. Okay, so that's all that I have for you on natural selection. The rest of what I'm going to talk to you about today is about the evidence for evolution. So far we've just been talking about processes and that kind of thing, like what it looks like and the definitions and so forth. And when we get into the evidence for evolution, things get kind of interesting. Your book quotes Pope John Paul II in 1996. He had something to say about evolution. And many religious leaders around the world also have these beliefs. In the United States, we are a little strange because so many of us do not think that evolution has any evidence and therefore are quote unquote believers in it. So I just wanted to share with you this idea that there are many people of sound religious faith that do in fact think that evolution has a lot of good evidence. So this is what Pope John Paul said. He said, "It is indeed remarkable that this theory, evolution, has been progressively accepted by accepted by researchers following a series of discoveries in various fields of knowledge. The convergence, neither sought nor fabricated. He's saying that all of these different fields that I'm about to talk to you about were not deliberately sought. There's no conspiracy, there's no lies. So neither sought nor fabricated. Of the results of work that was conducted independently is in itself a significant argument in favor of this theory. So 1996, folks, that's when the Catholic Church started to really publicly acknowledge that evolution is real. So after Pope John Paul came Pope Benedict, and you might know that Pope Benedict was a little bit more conservative than Pope John Paul, but this is what he had to say about evolution. He said, "I would not depend on faith alone to explain the whole picture." And he said, "They, religion and science, are presented as alternatives that exclude each other." Then he said, "This clash is an absurdity because there is much scientific proof in favor of evolution, which appears as a reality that we must see and which enriches our understanding of life and being and such." So, the current Pope has also has said strong statements about evolution. So, maybe that for those of you who are kind of skeptics about evolution, maybe this will help open your mind a little bit. But even so, even if it doesn't, we're going to talk about this, what both popes were talking about, the evidence for evolution. And it turns out that there are 5 primary lines of evidence for evolution. There's the fossil record, something called biogeography. Then there's comparative anatomy and embryology, molecular biology, and laboratory and field experiments. And those are the things that we are going to talk about for the rest of this lecture. Starting out with the fossil record, there's fossils everywhere. We saw about Cuvier and his fossils and so forth. And in Darwin's time, it was assumed that the deeper down the earth, in the earth a fossil was found, the older it was. And that's kind of like something that we still understand today. This because and Lyell, he contributed to this. We understand the geological processes. So older rocks are laid down deeper down, and newer rocks are laid down higher up. So that's called relative dating, and that's all very well and useful, but we've got some technology under our belts now, and we can now use something called radiometric dating. And so radiometric dating uses principles of physics to help us understand how old things are. I'm going to give you a little bit of background on this and understand that this gives us a quote-unquote absolute age. That doesn't mean that we absolutely know the exact age. What we're saying here is that we can put a date on things, an approximate date on things. So the way we do this is by using something called radioactive isotopes. And radioactive isotopes don't stay radioactive forever. They actually decay. And they decay at a very constant rate. And that constant rate is called the half-life. So if you have a radioactive material, when half of that radioactive material becomes stable or non-radioactive, and radioactivity is by definition unstable. That's why it's radioactive. So if that happens, then we have half of the material being radioactive and half of the material being non-radioactive, or we say stable. So that time that it takes half of them to go from radioactive to stable is called the half-life. So radioactive isotopes, they're constantly breaking down in this rate called the half-life. Nothing can change what the half-life is. There's a lot of bad science out there talking about this, whatever. Nothing can change it. And by measuring the relative amount of radioactive isotopes that are left over, we can actually compare that radioactive to the non-radioactive, and we can get a date. We can calculate a date on how old those fossils are. So that's kind of awesome. We also, when we're looking at fossils, we look at things called transitional fossils. And in one of the labs we'll be talking about transitional fossils. And transitional fossils are simply any fossil that shows a transition or kind of an intermediary between ancestral states. And so the amazing fossil that I want to talk to you about is called Tiktaalik. This fossil was discovered not that very long ago up in Canada, in northern Canada. And it was found in rock that was about 375 million years old. Now, the thing about Tiktaalik is that if you look at the bones in its feet, you can see some really interesting patterns. And we think Tiktaalik is a transitional fossil between the lobe-finned fish and the early amphibians. And early amphibians are ones that are spending part of their time on land and part of their time in the water, where there's literally like half and half of their time. Eggs in the water, but living on land, mostly full time. So Tiktaalik's in the middle, and we can see this looking at the bones in the fins of a lobe-finned fish. We see this really interesting pattern, and I'll be talking about this pattern multiple times. So what you see here is a bone that is connected to the body. This is the body up here, and this fin is connected to the body. So we've got one bone right there. And then connected to this one bone are these two bones. Always those two bones connected to this one bone. And then coming off of those two bones, we have a bunch of little bones. And that's the pattern that we see in the lobe-finned fish. And before Tiktaalik was discovered, we had the fossils of early amphibians, and we see the same pattern of one bone connected to the body, and then two bones connected to the one bone, and then a whole bunch of little bones, and these ones look very much like fingers connected to those two bones. If you look at Tiktaalik, you see a transitional fossil. You have that one bone connected to the body and those two bones connected to that one bone, but you start to see things that look very much like fingers. And that's one of the reasons why we say that Tiktaalik is a transitional fossil. It's showing a transition between the fishy ancestor and one of our ancestors, the early amphibians that came after the fishy ancestor, and Tiktaalik happened in the middle. So we see transitional fossils in all kinds of organisms, and this is not from your textbook, but I think it's really interesting that we see transitional fossils in horses. And I'm just going to really quickly step you through this. Your book does have a very nice discussion of horses and fossils, but this one is a little bit better, I think, because it really shows the transitions. So we're going to start. Oh, sorry about that. So we're going to take a look at the feet of these horses, or these horse ancestors. This first one is Mesohippus, and if you look at the feet of these Mesohippus, what you see are kind of three little fingers kind of sticking down here. These horse ancestors were pretty small, and they walked kind of on their tippy toes. And you can kind of see how those are actually toes. These bones go all the way up to here, where this is like knuckles or your wrist. If you look in Merychippus, you can see that middle finger that's kind of getting bigger, and the side fingers are getting smaller. And if you look at Pliohippus, you can see that's even more extreme. Now it's hard to see in this picture, but you can see those two little chunks of fingers kind of fusing right here. And eventually you get the kind of classic modern horse. So transitional fossil, you would say that Merychippus is a transition between Mesohippus and Pliohippus. And you could say that Pliohippus is a transitional fossil between Equus and Merychippus. Hopefully that makes sense to you. So your book, I said, has some nice fossils on horses, but it doesn't really show the bones. It does show what we think that they look like. And it's the first time that we are introduced to this concept of this tree, this branching diagram that's called a phylogeny. We'll hit that really hard in chapter 10. So this branching diagram is showing you how we think the ancestors of these organisms are. There's Merychippus, there's the modern horse, there's Miohippus, there's Mesohippus, and so forth. So even based on this diagram, what you can see is that Miohippus is a transition between Mesohippus, which goes further back in time, and something that's more modern, more recent in time, like Merychippus. So we'll be getting to know these diagrams really well. In the meantime, let me just show you something important, which says that this is the axis of time right here, and this is a graph, so it's got another axis right here, and that's the axis of divergence. We'll talk about that more later. The last question that I want you to think about with fossils, and this is our first line of evidence, is are there living fossils? You know, you might hear frequently that sharks are living fossils, and that always gives the impression that things have stopped evolving. But pretty recently, and if you're interested, I don't know if you could click on that link, probably not, but there is an article that you can listen to that talks about this really, really cool set of bones here that were discovered. There's a shark ancestor, and it turns out that the bones of the shark ancestor in the jaw are very different from the bones of the jaw of a modern shark. So even though the sharks look very, very similar to how they did, millions of years ago, they have in fact still evolved. The bone structure is very different, even though the external structures look very similar. Okay, so we're moving on to our second line of evidence, and this is biogeography. And biogeography is a little confusing to people who haven't heard about it before. So bio meaning life, right? And geography meaning like physical, like land structures and the way things are located around the world. So what biogeography talks about is how there are these geographic patterns that reflect the evolutionary history of organisms, because organisms live in places, right? So it's just the study of how organisms are distributed throughout the world. And interestingly, and very importantly to understanding biogeography, is that organisms that are more closely located together, the more closely related they are. So location means that that the organisms around there are most likely more closely related. Sounds a little strange at first, but if you think about it, most people who go to school with you have family nearby, and many of the people who are in your class have parents, grandparents, great-grandparents who are from this area. And so that's the kind of thing that we're talking about. Animals don't move around as much as humans do. So what we have here in our example of biogeography of the Hawaiian honeycreepers is that these three birds that look very different from each other live on different islands, but they're close islands. And these three birds are in fact physically close to each other. And even though they look crazily different, they are closely genetically related as well. So each of them has adapted to the different circumstances on the different islands. And for example, this one's drinking nectar and it lives in a very, very wet habitat. This one, the Maui parrotbill honeycreeper, lives up the slopes of one of the volcanoes and is eating insects. This one's drinking nectar, as I said before. And this one, it lives on this tiny island over here, wet forests, but they're mostly eating arthropods, spiders, caterpillars, that kind of thing. And you can see their beaks are really well adapted to those different eating habits. Okay, so location matters. What else matters in biogeography is that are there lots of different habitats available? Because if there's lots of different habitats available in a small location, you're going to get lots of different species evolving. It also matters who arrived first to that habitat. The bird ancestor, which is kind of shown here as the mainland finch, a North American finch, is really closely related to these. Now, it's not saying this particular finch is the ancestor. It's saying that they share a common ancestor with these Hawaiian honey creepers. So we think that a North American finch family maybe got lost on its way or somehow made its way to Hawaii and went through a lot of speciation once it got there. If there's lots of different habitats, then whoever arrives first is going to have lots of resources, and they got a good chance of adapting to use those various resources, and they're going to evolve. Remember, though, it's important. It's not the individuals that are adapting. Think about those rules of natural selection. It's the populations that are adapting. And so the populations are kind of dividing up and using different resources. Eventually, with enough time and habitat differences, you're going to get a wide variety of species due to biogeography alone. Your book does a really cool job of looking at these animals from around the world and trying to get you to understand biogeography. And I'm going to do a brief version of this. We're going to look at these 6 organisms. This is all 3 of these, by the way, are Australian marsupials. And all 3 of these are placental mammals that live either in North America or Africa. Okay, so these Australian marsupials, they all give birth in a very specific way that does not involve a big placenta, and all of these have placenta. Okay, so if you look at the sugar glider, they look a lot like the flying squirrel. And you might, by looking at them, just think that they're really closely related, and you would be wrong. A numbat looks a lot like a giant anteater. They eat the same kind of food. They eat ants and termites, and they have these long fused jaws. They don't really have any teeth, and you'd think that they might be closely related, and you'd be wrong. Same thing for this Tasmanian wolf and this gray wolf. They both hunt in packs. They're both top predators, but they're not that closely related. So who is closely related? And it turns out, I hope that you understand kind of obviously, that it's all the marsupial mammals are really closely related to each other, and all the placental mammals are more closely related to each other. I literally mean that this flying squirrel has more in common with this gray wolf than this flying squirrel has in common with the sugar glider. Yes, I literally mean that. And if you understand biogeography, you understand that it's because of location. These guys all had a common ancestor that was a marsupial, and they evolved into these different niches, these different habitats, and they all have a fairly recent common ancestor. Now, they're all mammals, so they all are related to each other, but not as closely related to each other, these two, as these two are, or these two, or these two. So, biogeography. The next one that we're going to talk about is comparative anatomy and embryology, and this stuff is really cool to me. So what we know is that many vertebrate embryos, soon after the egg meets sperm, right, you go and become the fetus and the embryo, they go through stages of having structures that are completely useless to the embryo and that are not found in the adult form, but they are found in the embryo. And the question is why? So let's take a look at comparing the anatomy of these embryos and trying to get our minds around, get a handle on what's going on here. So, okay, starting out with our shark. And there's 2 features. There's gill pouches and there's a bony tail. And sharks have tails as adults, and sure enough, they have tails as embryos, and that makes perfect sense, right? Sharks have gills as adults, and sharks have gill pouches as embryos, and that makes perfect sense. Those gill pouches are eventually going to develop into gills. Totally makes sense. When you start to look at a turtle, you can see that turtle embryos have tails. And again, this totally makes sense because, of course, turtle adults have tails. But here's where things get a little wonky, because turtle embryos have gill pouches, but they do not have gills as adults. They have lungs. So why does this embryo have gill pouches when it doesn't have gills as an adult? And no, it is not because the embryo uses that structure to breathe or to do gas exchange. Not at all. In fact, the only reason that this turtle has a gill pouch as an embryo is because it inherited the trait from its ancestor that happened to have gill pouches. And that's just weird. Let's look at this chicken with this really long tail. Now, I don't know if you know anything about chickens, but grown-up chickens, adult chickens do not have really long tails. So that's kind of weird. We've got this long tail here as an embryo, and a short tail as an adult. And sure enough, there's the gill pouches that once again are not functioning at all in the gas exchange like you might think. It's just not doing that at all. So finally, we have humans. And with humans, you see in the embryo, you see a tail. And I hope you know that at least most humans do not have tails. I mean, we have this thing called a tailbone, right? But a tail is defined as a bony protrusion that is extended out beyond the anus. Yeah, I mean, you gotta say what it is. That's kind of the definition of a tail. So in the embryo of humans, we've got this long structure that is a tail. But in adult humans, it's not there. It's inside the body. There's this little leg bud right there. And of course, there is the gill pouches. So why do humans have gill pouches? Because our ancestors had them and we still have the genes for them. Interestingly, remember that natural selection can only act on what's present and modify it. So what's happened to humans and some of these other organisms is that these bones kind of start to be co-opted, and they're useful in other structures, including the ear. So some of these structures of the gill pouch become part of your ear. The term here that I want you to look at is homologous structures. We've heard this word homologous before, and we heard it when we was talking about homologous chromosomes. And remember that homologous chromosomes were those chromosomes that were kind of similar to each other. They had similar information, but they weren't exactly the same. That's the same thing here for homologous structures in these embryos. And we can see the same kind of homologous structures in the adults. So we're doing comparative morphology here, comparative structures. And what you see here is we're looking at the limb structures of these 4 animals, and we're going to just compare the structures. Again, homologous structures are defined as structures that are present because a common ancestor had those structures. They're kind of similar, but that's only because the common ancestor had them. So if you look at the human arm, what you know is that there's one bone here that connects to the main part of the body, and then there's 2 bones here that connect to that one bone, and there's a whole bunch of little bones here that make these fingers. I hope this sounds a little familiar to you, because this is exactly the same pattern of bones that we were looking at when we were looking at Tiktaalik and the lobe-finned fishes and the early amphibians. If you look at horses, you have the same pattern. One bone here. These are actually 2 bones that are fused together. A whole bunch of little bones there, but only one set coming off of that. And if you remember back to when we were talking about the evolution of horses, and I talked to you about how those bones started fusing together in the fingers, that's what you've got. In bats, 1 bone here, 2 bones that are not fused up there, but you can see that they're fused down here. A whole bunch of tiny little bones, and then the fingers. And these fingers are quite elongated and very delicate and thin. Even porpoises, these like dolphins, have the same bone pattern. This is kind of weird. 1 bone connected to the body, then those 2 bones, many little bones, and yes, little tiny finger bones. They're not so tiny, but they're like really sturdy, and they help support that fin. You should know that fish, and we're not talking about porpoises here, or dolphins, or whales, or whatever, we're talking about fish. Only the lobe-finned fish have this pattern. The ray-finned fish, the vast majority of most fish on the planet, don't have this pattern of bones, just the lobe-finned fish, which is why we think that we share a common ancestor with the lobe-finned fish. The next kind of structure that's important to think about are vestigial structures. So that word is vestigial. And a vestigial structure is an extreme kind of homologous structure that sometimes comes to have little or no function to them. So we think about them as evolutionary leftovers because the structures had value in the ancestor, but the structure doesn't have that function anymore, or it has maybe a very, very reduced function. Your book does a great job of an example here. Vampire bats. The only thing that vampire bats eat is blood. Yes, it's real. It's true. Vampire bats drink blood. But strangely, and they have these huge front teeth, that's awesome to help them drinking that blood, cutting the skin of this, animal here to be able to drink the blood. But vampire bats have molars. And as you know, molars are for chewing. So grinding food. So what, really? Like, why would a vampire bat have molars? Well, they have molars because their ancestor had molars and they inherited the genes for these molars. And they don't use these molars. If a mutation comes along and kind of prevents a vampire bat from developing a molar, then maybe that trait will increase in the population. But it's not like they're just going to go away due to disuse. In fact, that's what Lamarck thought would happen. Remember Lamarck from way, way, way back at the beginning of this lecture? Lamarck was wrong. It's not about use and disuse. It's about the variation and traits acting upon that variation. All right, well, hopefully vestigial structures make sense to you at this point. So we have a little bit of a problem when we start thinking about evolution, because some of the structures that we look at are just like I talked about. Like you've got homologous structures, really nicely shown here, and we can say that homologous structures occur because you inherited this structure from your ancestor and natural selection acts on it to make it a little bit different. And that's all very well and good, but it turns out that some structures that exist in the world are not inherited in this way. Some structures are called analogous structures, and these structures, like the wings of an insect and the wings of a bat, these structures evolved completely independently. They didn't evolve from an ancestor who gave, genes to make wings. No, they're made of completely different starting materials. So even though they perform the same function, they evolved quite differently. We call it convergent evolution because the structures converge on a similar appearance. It's not like they're mating with each other. They just converge on a similar appearance. And so analogous structures like wings in insects and wings in bats are similar solutions to very similar problems, but they're built very, very differently. And they did not inherit that from a common ancestor. Okay, we're almost done with our lines of evidence. We just have a few more slides to get through. So let's go ahead and power through them. The second to last of our lines of evidence, the fourth line, is called molecular differences. And this is a very powerful, powerful tool in evolutionary tool belt for understanding how evolution works. So we know that if you look at your own family, that you have more DNA in common with people that are closely related to you in your own family compared to people who are less closely related to you. I mean, that's just a given. You have a grandma in common with your cousins, you're going to have more DNA from your grandma than you are from some random stranger who lives halfway across the world. So that's kind of the way it is with animals. Or organisms, you just kind of have to multiply it times a huge extreme, because although these organisms do share a common ancestor and they have DNA in common, the longer ago that common ancestor was, the less DNA they're going to share in common. So we're going to look at the DNA that is used to make amino acids in the the molecule hemoglobin. And so hemoglobin in humans has 146 amino acids in it. And so if you could know every single amino acid in this chain. If you compare human hemoglobin to rhesus monkeys, which are a very close relative of ours, they have 138 of the exact same amino acids, 138 of them, with 8 differences. It's still blood, still hemoglobin, it still does the same exact function, but it does it a little bit differently. We've got a little bit of differences there. So what about a dog? I mean, clearly a dog is less closely related to us than a rhesus monkey is, and it turns out that a dog only has 114 of the exact same amino acids, and there's 32 that are different. Birds have 101 of the exact same amino acids, and 45 of them are different. And then if you get as far away from humans as you possibly can, but still be a vertebrate, still have a vertebral column, you might look at this fish called the lamprey. Lampreys don't even have jaws, but they do have a vertebral column. They've got teeth, but they don't have jaws. It's kind of weird. And they have 21 amino acids in common with us and 125 different in that hemoglobin molecule. So just by comparing these, I mean, I guess we already knew that humans and rhesus monkeys were more similar, and that dogs are mammals, so of course they're going to be more similar to us than birds. And of course birds are, tetrapods. They've got 4 limbs, and they're going to be more similar to us than lampreys that don't even have a jaw. The really cool thing about looking at molecules is that you can use them to put a date on things. So by looking at these amount of differences and understanding how fast mutations happen and so forth and so on, and comparing it to the fossil record, we can say that humans and rhesus monkeys had a common ancestor about 23 million years ago. And that humans and dogs had a common ancestor about 46 million years ago. These common ancestors were neither humans nor dogs. They were something that led down one path towards dogs and one path towards humans. We can say that birds and humans had a common ancestor about 112 million years ago, and that humans and these just strange creatures called the lampreys had a common ancestor 564 approximately million years ago. So again, a very, very powerful tool, and hopefully you're reading up on that. Our last line of evidence is the experiments in evolution. So our first experiment is not an experiment at all. It was actually something that is kind of done accidentally, but the really cool thing about this is it's been done accidentally, but repeatedly done accidentally. And that is that the way that grass is cut on a golf course is always, pretty much the same. You've got grass in the rough, and that's only cut once a month. You've got grass on the fairway that's cut, about 4 times a month. And then you've got grass on the putting green that's basically cut every other day except for the weekends, because people gotta golf. And what scientists have found, and this is the really cool thing, like all of this patterns here, cutting grass, that's just very, very common. But all of this grass actually started out as very, very similar. But it has evolved because of these different cutting regimens, and because grass has to reproduce by making seeds, that the grass in the rough is a very slow at sexually maturing and is very low in seed production. Why should it mature fast? It's not being cut except for once a month. There's no reason to force it to reproduce sooner. And yes, grass has to make seeds in order to reproduce. So the ones in the fairway, those are kind of slow to mature sexually. Again, that's making the seed heads. And they don't make a lot of seeds, but it's more than the seeds in the rough. And if you look at the seeds, the grass in the putting green, they're sexually maturing very, very fast. In other words, if they don't sexually mature within a couple of days, they're not going to be able to reproduce. And so they're becoming sexually mature very, very, very quickly. And they have a very high rate of putting out seeds. If you ever go onto the putting green or if you ever play golf, you should take a look at this. It's really, really cool. We have forced the evolution of putting green grass. The last thing that we're going to talk about is way more unfortunate, and it's it's an experiment in quotes. We didn't mean to do this experiment again. Like we didn't mean to do the grass seed on golf courses experiment. It just ended up being this way. And again, not such a good thing because bacteria also evolve. And back in the 1940s, people would frequently, this is how you kind of test this, you'd put bacteria in a petri dish, and if you put down a little disc covered in penicillin, what would happen is that the bacteria, in this case, the bacteria is called Staphylococcus. The kill zone, that's the distance between where the penicillin is put in and where the Staphylococcus can actually live. So this whole region here is called the kill zone. You can see how big that kill zone used to be. And as time went by, as we started abusing our use of antibiotics, the Staphylococcus became more and more able to resist that penicillin. Now, I want you to remember that it's not evolving because it needed to. It's evolving because some of these right here, like some of these bacteria right here that were a little bit closer, right, they still got to reproduce. And so the ones that were killed off, the ones that were completely killed off, those did not reproduce. But like that guy right there, that totally reproduces. And so it's just a little bit closer. And so that's the one that survives to reproduce. And so you get a little bit closer. As time is going by, this guy right there, that little guy right there, that's the one that's reproducing. All these other ones are dead in here. But that one survives and it reproduces, and it's going to be the kind of the parent of the guys today. And so what we know today is that you basically can't kill Staphylococcus bacteria with penicillin. I mean, even forget about you being allergic to penicillin or whatever. A lot of people are. Staphylococcus is not killed by penicillin, and it's because we have selected unknowingly, we have selected for the bacteria that was resistant to penicillin. Those are the ones that survive to reproduce. So yeah, not such a good thing. But I hope you can see why it's so important to learn about evolution. These things are happening, and if they're happening, you should probably know about it. And hopefully I convinced you. I know I didn't convince everybody, but it's still important to know. All right, well, see you in class.